Good evening, Vanshika. Okay, so I'm Rahul Saha, and today we'll be talking about more about the female reproductive tract. Like we had talked about the syncytiotrophoblast, like little bit. Also, we'll go deeper into that. In the last class, I had shown you a diagram, which I'll share again and just rewind the things for you. Yeah, just share the diagram again. Okay, now you will be able to see the diagram. In just a few seconds, you'll be able to see it. Yeah, this diagram only I was talking about. So I have told you that this is the specific blastocyst, the blastocele, and you have the inner cell mass, you have the trophoblast, and this comes in contact with the epithelial cell layer, the apical layer of the epithelial cell, which has got, you can say, finger-like projections, microvillus projections, and then it expresses mucin 1 also, and after it comes into contact, after the specific blastocyst comes into contact with the epithelial cells, the mucin expression is reduced because mucin is a mucous membrane, produces the mucus layer. It is anti-adhesive, so it has to be reduced because need to adhere this blastocyst with the specific, you can say, the layer of the epithelial cell of the endometrial layer. Okay, and then they have the expression of L, you can say selectin, and then various kinds of integrin. Okay, and these all come under the category of cell adhesion molecules. You have troponin also, or trophinin, which is expressed by the blastocyst only. One more thing that I had shown you, or that I had made a point about, was you can see the embryonic pole or the animal pole coming into contact with the epithelial cell layer, and then it will penetrate into the stromal layer, which is the connective tissue layer of the endometrial lining, right? So why not it happens the other way around? Why not this layer comes into contact, the unembryonic or the vegetal pole, with the epithelial cell? In every successful pregnancy, only the embryonic pole comes into contact, right? The reason behind it is still in research, but the scientists have a hypothesis that the trophoblast cells at the embryonic pole is different, little bit different, which compared to proteins, expression level of the proteins, epigenetic regulation, with the, or if you compare it with the trophoblast cells present at the, you can say, aembryonic stage. Therefore, it will get through, and then this epithelial cell will reseal the gap as if it has been engulfed or swallowed. Okay, so this is known as a complete implantation process after the blastocyst hatching has taken place, means the zona pellucida is digested. Now, this process is known as implantation process. Okay, we keep it very clear in your head. Okay, this is known as implantation. You can see, I'll show you other images also. You can see this is 5 to 6 days, 7 to 8 days. Okay, so all these things will be occurring, okay, in during the implantation process only. So I'll explain the other diagrams also to you so that it becomes pretty clear. Okay, so if you see the other diagram, okay, of the implantation process, so the next stage of implantation, when we talk about, that diagram is there in front of you, is penetration of the uterine epithelium. Okay, in primates like us, the cellular trophoblast undergoes a further stage in its differentiation just before it contacts the endometrium. Okay, so in the area around the inner cell mass, just the area of the, you can say, blastocyst around the inner cell mass, the cells derived from the cellular trophoblast, okay, which are known as, you can say, cytotrophoblast, they fuse to form a multinucleated, okay, a multinucleated syncytiotrophoblast. Means the cytotrophoblast, they fuse with the you can say endometrial epithelial cell to form syncytiotrophblast layer. Okay, now only a small area of syncytiotrophblast is evident at the start of implantation. Okay, now this structure soon surrounds the entire embryo, means the covering that I told you is this covering only. Okay, if you see the image, is this covering only. If you look at the covering, this covering is that only. Okay, the syncytiotrophblast one you can see here, syncytiotrophblast layer. Okay, the fusion layer is syncytiotrophblast layer, and the syncytiotrophblast is covering the entire embryo. You can see the syncytioblast covers the entire embryo. Okay, that is a great stuff that is happening. Okay, that is known as syncytioblast formation. Now, small projections of syncytioblast, what happens is they insert themselves between the uterine epithelial cells. Okay, between the uterine epithelial cells, they insert themselves. Now, what is the consequence of it? You can say they spread along the epithelial surface, okay, of the basal lamina that underlies the endometrial epithelium to form a flattened trophoblastic plate. You can see here, if it is given, they form a specific, you can say here, a trophoblastic plate is formed. Okay, trophoblastic lacuna that is formed, trophoblastic plate, then it is formed later on, and then within a day or so, the syncytoblastic projections from the small trophoblastic plate begin to penetrate the basal lamina. Okay, and then what happens is you have specialized cells. Okay, like there is an invasion of endometrium by facilitated, you can say, lytic enzymes. Okay, and in addition to this, the invading blastocyst induces, you can say, cell death of endometrial cells. That is known as apoptosis. The endometrial cells will undergo cell death. And why will the cell death happen? In order to increase the available space for the embryo. Otherwise, if the cells are there, how will you get in? Right? So that is, the cell death is happening. Okay, that is again a reason how this is able to get through, because the cells here, it will otherwise burst open, right? It is getting implanted, so you will be having cell death here in the endometrium, induction of specialized cell death. Okay, so that thing will be occurring, cell death will be happening. Now after this, what will happen is there is also an evidence that syncytioblast of the early, you can say, embryo is little bit different. Okay, is a variant, you can say, from which the, you can say, placenta comes out, or placenta is formed. Okay, so the early implantation continues projections from the invading syncytoblast. They envelop the portions of the maternal epithelium, you can say endometrium, and the blood vessels, and they will keep on eroding the blood vessels. Okay, and maternal fluid begins to fill the isolated, you can say, space that have been forming in the trophoblast. That is happens, and that isolated space only is technically called as this one, lacunae, trophoblast lacunae, because here the cells are dying. That is known as trophoblastic lacunae. Okay, here they are being, you can say, filled by the maternal fluid, and that in the end gives rise to, you can say, amniotic fluid also. Okay, the trophoblast will change the character, and then it doesn't invade that much, it won't go deeper and deeper, it will stay there. And at this stage, you can have something known as spotting also. The female, that is not a menstrual cycle, but leakage of blood from the uterus at this stage is called spotting, which is sometimes misinterpreted as an abnormal menstrual period. Okay, so when the embryo burrows into the endometrium and some, you can say, cytotrophoblastic cells fuse to form syncytoblast, you can say, the endometrial basal, you can say the fibroblast like cells, the stroma, which are the connective tissue, okay, they also swell, okay, because they will provide the much needed glycogen and lipid, okay. These cells are known as decidual cells, okay. You might be seeing here, these cells are known as decidual cells, okay. uh, the decidual cells here and there, okay, these cells are known as decidual cells. They swell, okay, these are the decidual cells you can think of, okay. This is the amniotic cavity that will be filled, okay. So that is what happens in case of mammals, okay. Now this entire decidual reaction happens and few cells they become the decidual cells, okay, completely. And an embryo, again, one more thing is very important. If you give proper, you can say, attention, an embryo is again antigenically different or not to the, you can say, mother. It is completely antigenically different to the mother. It is not same as the mother. Just like sperm was antigenically different to the, you can say, the man, right? So we had the, you can say, Sertoli cells, they produce the testis blood barrier so that the TH cells they do not infiltrate and they do not recognize, and one doesn't mount offense or immune response against the sperm because they have undergone recombination event. Okay, homologous recombination is known as crossing over at the packaging. Now they will produce a little bit of different proteins which will be recognized by our own immune system, okay, as antigen or as a foreign antigen. Thereby the immune system will mount an offense, okay. So I'll write it down for you. So then our immune system will mount an offense. So you can say an embryo is antigenically different to the mother, right? Now, since an embryo is antigenically different from the mother, and consequently, what should happen? It should be rejected by the cellular or cell-mediated immune response that you might have studied in chapter 8. Okay, similar to the reaction which should have happened there also, like in case of the sperm, or in case of some kind of transplant, like if you have gone through a liver transplant, a heart transplant, you can say kidney transplant. But the thing is, a primary function of the decidual reaction. Decidual reaction means, you can say, the decidual cells are tightly adherent and they form a massive cellular matrix or cellular, you can say, structure that first surrounds the implanting embryo and then later occupies most of the endometrium. That is the role of decidual cell. So decidual cell, what they'll do is they will adhere to one another, okay, tightly. Always no, decidual cell will adhere to one another tightly, and like this they will first, you can say, adhere, and then they'll be, you can say, coating or surrounding the embryo like this. So this is the decidual cell layer. This is the decidual cell layer adhering to it tightly, okay. And concurrent with the decidual reaction, okay, the leukocytes that have infiltrated the endometrial stroma before the decidual reaction, some leukocytes means white blood cells are only T cells, lymphocytes and all. Some leukocytes would have infiltrated the stromal layer, okay, during the during this phase only, okay. They secrete a specific chemical. You might have learnt in the immune system response known as cytokines, specialized interleukin. They secrete interleukin 2, which prevents maternal recognition of the embryo as a foreign body during early stages of implantation. So those leukocytes, they help out the decidual cells to prevent maternal recognition of the embryo as a, you can say, foreign body. So the leukocytes which might have infiltrated here, they'll secrete a cytokine, right? You might have studied the about the cytokines, the cell mediators or the cell secreted mediators. Okay, so one of those is IL-2, interleukins. Okay, interleukin 2. This interleukin 2 prevents the mother from recognizing the recognizing the embryo at its early implantation stage as a foreign body. Okay, so the decidual reaction plus the IL-2. Here, understand the concept, the decidual reaction along with interleukin 2, they prevent the maternal reaction. They do not let the maternal reaction to take place. Okay, so still you can say this resistance, the adherence junction, okay, how does it, you can say, lead to complete abolishment of the infiltration of the leukocytes is still under intensive research. Okay, but the most accepted postulate is what I explained to you. Okay, frequently what all happens is a blastocyst, you can say, it fails to attach to the endometrium. Okay, and sometimes implantation then will not happen, it doesn't occur. So failure of implantation is a particularly, you can say, taxing problem in in vitro fertilizations. In vitro fertilizations, because if you fail to implant, you have gone for ZIFT, zygote interfallopian transfer or gamete interfallopian transfer, all that has happened, but implantation is being failed. Okay, so the thing is, the success rate of implantation of transferred embryos, okay, it remains at approximately 30 to 35% only. Okay, that is the thing. It is not very high. It always is 32. You can say 35%, because when you implant an embryo you might not have gone for capacitated sperm. Okay, then that again would not have undergone or gone through so many reactions that would have happened due to sperm capacitation and then acrosomal reaction entry. Okay, so that has been achieved in the lab only. So those things, they do make a lot of difference, because each and every signaling protein, it makes a huge difference. Okay, so I'll write down some basic stuff for you that I explained verbally. Okay, so the cells derived from the cellular trophoblast, that I'll write, the cells derived from the cellular trophoblast layer. Okay, they are known as, you can say, cytotrophoblast. They fuse to form, okay, these cells only directly they fuse to form a multinucleated you can say cell known as syncytotrophoblast. Okay, syncytotrophoblast. Trophoblast. Now, although a small area of syncytioblast is evident at the start of, you can say, in implantation, this structure will surround the entire embryo. Okay, this structure surrounds the entire embryo that I had shown you in the image. This happens at the animal pole only. It will surround the entire embryo during implantation. So that always remember it surrounds the entire embryo during the process of implantation. Now, the small projections of syncytioblast it will insert themselves between the uterine epithelial cells. Okay, always remember it will insert themselves between the uterine epithelial cells. They will keep on inserting. Okay, that will happen between the because the uterine epithelial cells are there and they will insert their open you can say projections between the epithelial cell of the endometrial lining and they spread along the epithelial surface of the you can say basal lamina and form a specific trophoblastic plate. Okay, and it will keep on invading. It is a very invading tissue. Okay, which erodes its way into the endometrial stroma. Okay, it will keep on eroding the entire epithelium because the thing is you should be thinking about the fact that how does it get through the epithelium. Okay, it is not a heavy machine gun that you will keep on you can say firing and putting missiles through, right? So what it will do is this is the epithelial layer. This is the epithelial layer. Okay. Now, how will the syncytotrophoblast here? It is going through, right? So the syncytotrophoblast, this layer multinucleated, it will break down this cell layer. It will break down this cell layer. Okay, so it is highly erosive in nature or highly invasive in nature and below the epithelial cell layer and between epithelial cell and the stroma, there is the connective tissue. I always told you, as per the anatomy, we always have something known as basal lamina. The same basal lamina it was known as or is known as membrana granulosa when we talk about the tertiary follicle or Jessel follicle, right? Between the you can say internal thecal cell and the follicular or granulosa cells. Here also basal lamina will be there. Basal lamina has reticular fiber. Okay, it has the lamina lucida, lamina densa. Okay, so all those things they have, but that will also be degraded. Okay, it will be able to degrade the epithelial cell and both basal lamina and reach the you can say stromal layer. Now, how will it be able to break? It is facilitated by okay lytic enzymes okay which is produced by the syncytotrophoblast okay that I told you also It is accentuated by, triggered by, triggered by lytic enzymes. Okay, some lytic enzymes are matrix metalloproteases, but those are not in your course. I'll just write lytic enzymes, and also it causes the induction of cell death. Okay, the induction of cell death or apoptosis or apoptosis in the or of the endometrial cells, of the epithelial cells of the epithelial cells of the endometrium. So that will be again a great, you can say, addition, because half of the endometrial cells will be killed by the lytic enzymes and some will be automatically dead, so it will lead to the, or its path will be completely clear to reach the, you can say, internal layer. Okay, so it will be able to reach the stromal layer after that. Okay, so the thing is, after the embryo has got implanted, okay, now we have completed the implantation process. Now after the embryo has implanted, okay, the cells of the inner cell mass, they diverge into two distinct lineages. Okay, one lineage is known as hypoblast, one lineage is known as epiblast. Always remember. So when the cells and when the implantation process is complete, okay, when the, then I'll tell you the specificity of chorionic villi that is given in the NCERT. The question comes a lot also in the board examination. Process is over, the inner cell mass, okay, is divided, the cells, they undergo differentiation into two different cell types, into two, you can call, lineages. Okay, into two lineages. The first lineage is known as the hypoblast. Okay, hypo means under, so hypoblast. Hypoblast is also known by the name as primitive endoderm. Now what they do is, these cells will form the extra embryonic cells. Okay, like the inner layer of the placenta, not the real embryonic structure. They will form the extra embryonic cells. If you remember, a few classes ago I had explained to you that the inner cell mass forms both the extra embryonic and, you can say, intra-embryonic or the embryonic structures. Embryonic and extra-embryonic both they form, right? That is a great neat question. And the trophoblast only produces extra-embryonic structures, those that do not belong to the embryo, like the outer layer and the middle layer of the placenta. And this will form the inner layer of the placenta, and the, you can say, both the embryo. So inner cell mass, how will it form both? So it will divide into two, hypoblast or the primitive endoderm, that will produce the inner layers of the placenta, and then the other one will be known as the epiblast, which will produce the cells of the embryo itself. Other layer is, or other lineage of cells are known as epiblast, which will produce the embryo. Okay, always keep this in your mind. It will produce embryo. Okay, now what happens is, how the hypoblast forms in human embryos is still not understood. Okay, it's still not understood, but various transcription factors, they play a role. Okay, nanog and all, but those are not in your course, so we won't go down into that detail, that how are they formed, which all transcription factors are turned on and turned off. Okay, but still they are not completely understood. A lot of things in the event of, you can say, developmental biology, this process is known as developmental biology. Okay, this field, that how do we develop, that is known as developmental biology, is still uncovered. Okay, it has to be unveiled. It is still under a veil, under a, you can say, guise, that you cannot, the scientists, they haven't been able to know which pathway is taken, which transcription factors are turned on, how and why. Okay, so lots of research is left to be done. Okay, so what happens is, that is how you develop these. Okay, so I'll just show you an image which will really excite you. Okay, so let me show you the image. We won't go very deep into it. I'll just show you a specific image. How does the body develop? I'll just show it to you. Now you will be able to see the image. In a few seconds it will come onto your screen. Now, yeah, you can see it right now, that this is the image that I wanted to show. Okay, just look at this image. Okay, so you have the inner cell mass here. Okay, trophoblast layer. Then you have the epiblast. This is the hypoblast. Okay, epiblast, hypoblast, and then you get the amniotic cavity. Okay, so you can see all those things happening here. Okay, this is being stretched. Okay, this becomes the primary yolk sac. Okay, so in parietal, you can say, endoderm, you can go down. Okay, so that is the coelom forming extra embryonic remains of the primary yolk sac. Okay, secondary yolk sac again forming. Okay, so after this will be the event. Primary yolk sac will be divided into two. Okay, so you can see all these events, they are, you can say, occurring. Okay, this and this. Okay, that is happening. So those are the things that really excite an individual towards taking the route of a scientist, of unraveling this developmental biology, you can say, state. Okay, but we don't have that much in our course that I can explain this entire thing to you. Okay, if the time permits in some offline class, because this is really interesting. How does the hand form, how does you, or how does the head form, the neurulation, gastrulation, the all three embryonic germ layers. Okay, so those things are there. I'll just show you one more thing. This is the entire, this one is the entire, you can say, the flow chart of how things proceed. Okay, you can see cleavage trophoblast, cytotrophoblast, syncytiotrophoblast. Then inner cell mass, they become epiblast, hypoblast, extraembryonic endoderm, yolk sac, endoderm. This will become amniotic embryonic epiblast. Then this will become embryonic primitive streak. That will become the neural crest, neurulation, then extraembryonic mesoderm, embryonic mesoderm, notochordal process, then embryonic endoderm, allantic endoderm. But those things are not, the description of these things are not there, but they're really, really, you can say, great to study, okay, as far as the interest level is concerned. So we'll stop it here and we'll then delve into the female reproductive tract, which is our next topic, okay. So this is all about our interest in the, you can say, developmental biology process, okay. Then we'll shift our attention to the female reproductive tract, okay. So when we talk about the female reproductive tract, okay, the female sexual characteristics are the, you can say, in standard terminology, the female primary sexual characteristics are the internal structures of the reproductive system only or not, right, including the ovary and the female sex accessory ducts like oviducts, uterus, vagina, okay. Now we have the external genitalia, okay. So the thing is, once we talk about the female reproductive tract, we cannot escape or evade ovary, okay. So the paired ovaries, also they are known as female gonads, okay. The paired ovaries, also they are known as female gonads, female reproductive tract. So the paired ovaries, they're also known as gonads, right? Gonads or ovaries are same only. They are oval shaped structures in the upper pelvic cavity. Okay, always know, anatomically they are oval shaped structures in the upper pelvic cavity. That is there. And you can say they are present against the back of the pelvic wall on either side of the uterus. That is there. Now, these small almond-shaped organs are white or yellowish in color. Okay, have a lumpy surface. You can say, so white or yellowish in color. The color I'll write. And the size also I'll tell you. Okay, so the mature ovary measures about 1 to 2 inches in length and 0.5 to 1 inches in width. 1 to 2 inches in length, 0.5 to 1 inch in width. Okay, so that is what the length and width of an ovary is. And they have two essential roles. We all know that: production of female gametes, that is oocytes or egg, and secretion of several hormones, including estrogens, progesterone, inhibin. Okay, so they do both kinds of specialized work. Now, the ovaries are also innervated by which one? By the autonomic nervous system, okay, and receive a specially rich supply of blood, okay. So that is there, and they are anchored in their place by specialized bands of or sheets of tissue known as ligament, which connect them to the nearby organs, okay. That is there. So those are not that kind of ligament that you have studied. They are just the folds of the visceral peritoneum, okay. That if you have studied proper anatomy in class 11th of the peritoneum, you will be able to know. So we won't go there into that detail about the visceral peritoneum folds and ligaments, okay. So that is a different thing. So you have, and then we'll come on to when we cut open an ovary, okay. When we cut open an ovary, and examine it under the microscope, okay, so the external surface of each ovary is covered by the surface epithelium. Okay, it is known as germinal epithelium. The external surface of the ovary after it is cut open. After it is cut open is known as germinal epithelium. Okay, germinal epithelium. Okay, because you can say it was thought that female germ cells were derived from this tissue only, just before. So historically it is termed as germinal epithelium, but it is now known that the germ cells originate outside of the ovary during the embryonic development. Okay, that is there. But earlier it was thought that you can say it originates from the epithelial layer of the ovary only. Okay, so underneath the tough protective epithelium is a protective layer of connective tissue. Okay, that is given in NCRT as the tunica albuginea. Okay, under the epithelium layer is a tough protective layer of connective tissue. Under the epithelial layer is a tough protective layer of connective tissue known as tunica albuginea. Okay, tunica albuginea. Now this connective tissue framework, okay, is divided into, that is known as the ovarian stroma, that I told you the stromal cells will invaginate and will get through into the formation of the Graafian follicle that will again divide into theca interna and externa. So this connective tissue framework, which is the ovarian stroma, is divided into a more dense outer ovarian cortex and less dense central ovarian medulla. Okay, so this you can say this connective tissue framework is divided into a more dense, that is the stroma only, more dense outer ovarian cortex, outer ovarian cortex and a less dense central ovarian medulla. Central ovarian medulla. Okay. Now the ovarian medulla, that contains large spirally arranged blood vessels, lymphatic vessels and nerves. Okay. It contains large arranged blood vessel supply, lymphatic vessel supply and autonomic nerve supply, lymphatic vessels and nerves as well. Okay, so that is there in the ovarian medulla. Now the ovarian cortex that contains the female germ cells. Okay, now each female germ cell or oocyte is enclosed in a tissue sac that we have already talked about. It is known as the ovarian follicle. Okay, follicle is Latin for little bag, you can say. It's a Latin name for little bag. So the thing is, always know that the oocyte is enclosed, okay, in ovarian cortex. Okay, the ovarian cortex is the main entity here. The ovarian cortex comprises of germ cells, not the medulla, okay, which most people make mistake in, and it is enclosed in ovarian follicle. Okay, primary, secondary, tertiary, or the Graphian. The follicle is the Latin name for little bag that I told you. And between the oocyte and the follicular wall, we have already discussed this also. We have the proteoglycan layer, the acellular layer known as zona pellucida, okay, which is mainly secreted by the oocyte. Okay, so we will not go into that detail. We have learnt about it already. Now, when we talk about the stages of follicular growth, okay, so the ovarian follicle is the basic functional unit of the ovary. So we have talked about the membrana granulosa, the granulosa cells, all those things we have talked about. So I'll just skip that part here, Graphian follicle and all. So that is there. So now we'll go for something known as ovarian steroid hormone synthesis. So we have estrogen, we have progestin, and we have androgen. So that also we have talked about, right? That how does they, or when does the receptor for the FSH get expressed onto the, you can say, granulosa cells, and how does the internal thecal cells they express the receptor for the LH, and then you can say the specificities of the LH reaction. Okay, the LH will be expressed, and then it is converted. The testosterone gets through the, you can say, channel, the gap junction. Okay, and then it will be able to be converted to 17 beta estradiol, because the aromatase enzyme is secreted by the granulosa cells, okay, which are the, which are expressing aromatase because of expressing the FSH receptor, and that will be able to induce the production of 17 beta estradiol, and 17 beta estradiol will then act upon the granulosa cells and make them express the LH receptor. Okay, and that will close those gap junctions. Okay, break the diplotin arrest, not let the cyclic GMP come in, cyclic AMP come in. Phosphodiesterase 3A will be activated, MPF will be activated. That is known as LH surge, and then you will have your ovulation. Okay, so that will be a great pathway that we should be knowing always. Okay, now we have the structure, so you don't no need to go for the structure of testosterone, progesterone, and estradiol. They are just, you should know, they are the derivatives of, you can say, cholesterol only. They are steroidal hormones, they are not proteins whatsoever. Okay, they are having long isoprenoid rings, benzene, you can say, chains also, isoprenoid chains also. So they are the derivatives of cholesterol only. Okay, now what happens is, two major one major thing, I'll just show it to you, which will explain the biochemical event. Okay, which will open up your eye to this biochemical event of menstruation, and then menstruation will deal in detail via, you can say, negative feedback loop in the next class. So I'll just share an image with you and explain this event. biochemical event, or how does this switch happen between estrogen production and progesterone production. Okay, that image will be visible to you in a few seconds. Okay, just it's coming on. Okay, now it has come on. You can just see the image. Okay, and just observe. You don't no need to, you can say, mug it up anything. You just observe. This is the tertiary follicle. Okay, before ovulation. This is the pre-ovulatory follicle immediately before ovulation, and in the corpus luteum also. So this is the theca interna. You can say these are the granulosa cells. So they have taken the delta 5 pathway. No need to know what is delta 5 in the real sense. What I'm showing it to you is cholesterol becomes, you can say, pregnenolone, then DHEA, then androstenedione, and this diffuses, and then FSH comes here, and FSH will produce aromatase. Aromatase will convert this into, you can say, testosterone to estradiol, and you can see here the estradiol is going into the blood. Okay, so this is what is happening. And thecal cells, I had told you, they express LH receptor. So LH receptor, LH action on, you can say, thecal cell, it is causing the various enzymes to be produced, which is taking this pathway from cholesterol. Okay, taking this pathway from cholesterol. Now LH receptor, which is being expressed on the, you can say, corpus luteum when ovulation has already occurred, that will lead to another pathway that is known as delta 4 pathway. Here ovulation has not occurred. Okay, that will lead to delta pathway, delta 4 pathway, that is known as cholesterol will be again taken, and sorry, it will be converted to pregnenolone. But you can see here it will be converted to, now pregnenolone gets converted to progesterone instead of 17 hydroxypregnenolone and DHEA. That takes a different route. Now that is why progesterone is produced from corpus luteum, because the biochemistry changes here. They take the delta 4 pathway, and then you produce progesterone that goes into the blood. Okay, and then progesterone is then converted onto estradiol again. But here you don't see any progesterone production. That is why corpus luteum is able to produce progesterone, because they take the delta 4 pathway. This is also LH, this is also LH. But here the biochemistry is different, because you have the oocyte inside it. Here you don't have the oocyte. So here LH has a different effect, a different expression of the enzymes which take the delta 4 pathway. Here LH has a different effect which takes the delta 5 pathway. That is the main difference that I wanted to show you, because people say that corpus luteum produces progesterone, but no one tells you the reason why. This is the reason why corpus luteum produces progesterone, and the Graafian follicle before ovulation does not produce progesterone. Okay, that is the main reason, delta 5 and delta 4 pathway. This is how you understand the concept behind the real biology or the biochemistry going on behind the scene, okay, which is really causing the event to occur. So I think this lecture is completely clear to you. In the next lecture, we will be dealing with the specificities of each and every cycle pertaining to the menstrual phase, and I'll be explaining it via the negative feedback loop, which directly is asked in the NEET examination. The looping process, the negative feedback loop, in case of the menstrual cycle and its hormones only. How does it get inhibited? How will it get upregulated, downregulated? Why the hormonal events? Okay. So if you had any queries, you can ask your queries on my personal number, and I'll be replying as soon as possible. Okay, then thanks a lot. Let's meet